Hello, I'm Octavio, and I'm going to be talking about anthrax. Anthrax is not a common disease nowadays, but it was prevalent in the past, and it had some considerably severe physiological effects. So, in this PowerPoint presentation, I'm going to be talking about the history of anthrax. I'm going to talk about the etiology, which is both just to give it some background. I'm going to talk about the pathogenesis, and I'm going to talk about the pathophysiology, which are the actual physiological effects of anthrax disease. Starting with history, anthrax originated in Egypt and Mesopotamia around 1500 to 700 BC. Its earliest recordings of anthrax were the boil-like lesions on Pharaoh's cattle. It's the first episodic outbreak, and the first outbreaks occurred in 14th century Germany and 17th century Russia and Central Europe. Livestock were the animals affected. However, it could affect humans, but it was not recorded during those times. The first clinical descriptions of anthrax were given by Merritt in 752 and Fournier in 1769. However, Fournier was actually the one that discovered or identified the source of the disease being from the animal industry, whether that be meat, skins, wool, any sort of animal product. In the 1800s, early researchers made the association between the anthrax disease and the presence of rod-shaped bodies, eventually referred to as bacteria, and then Bacillus anthracis, which is its scientific name. But before this, actually, early doctors spotted the link or made the first association between the disease and the animal hair industry. And this is what prompted early researchers to figure out what was actually causing the disease. And this is how they figured out that it was rod-shaped bacteria. And also, it should be noted that anthrax was not always called anthrax. It was used to be called wool sorters disease. And the reason why is wool sorters, which were people that sorted wool and other animal products, they would get this pathogen by touching contaminated animal products, or like I said, wool, skin, meat. And so it was called wool sorters disease as a result. Continuing on with history, in 1877, microbiologist Robert Koch conducted breakthrough research on Bacillus anthracis. He is actually credited as the pioneer for microbiology, especially anthrax disease. He discovered the anthrax bacteria formed spores and were resilient. And by resilient, I mean they were very robust. They were able to, survive harsh environments. They were able to uh, they had mechanisms that kept them alive during the points where they couldn't germinate. He outlined the life cycle of anthrax bacteria, so how it goes through its phases, with either within the body or just in general. Uh, he determined that there was a relationship between the microorganism and the disease through his studies of anthrax. And that's how what made him create the Koch's postulates, where what I just previously said, the relationship between a microorganism and a disease. In 1881, microbiologist and chemist Louis Pasteur created the first prototype vaccine for anthrax. And although that vaccine was not for humans, it was mainly for livestock. But later on in the 1950s, the first human vaccine was actually created and released. And then it was improved upon in the 1970s to make the vaccine we currently use today for anthrax. Uh, Last slide for history. During World War One in the 1900s, anthrax was used as a biological weapon, and it set the stage for biological warfare. That's why biological warfare is banned, because of when the German and Japanese forces exploited anthrax to kill their opposition. It was primarily used to kill soldiers and citizens, but was also used to kill livestock being traded to Allied forces. In 2001, anthrax attack takes place after events of 9/11. Now, this is the most current progression of anthrax. After this, it hasn't been that common, but it is notable that it can be spread and it can be used as a weapon. Moving on to etiology. Anthrax is an infectious disease caused by rod-shaped bacteria, Bacillus anthracis, as previously stated. Bacteria is naturally found in soil, plants, and water, and of course, the water, soil, and plants are all contaminated with the actual bacteria. It affects domestic and wild animals when they inhale or ingest anthrax spores. So these spores are the things that are going to be found in soil, plants, and water. And the spores are basically the mechanism that the bacteria use to protect itself until it reaches an environment where it can germinate. Human transmission happens when humans come into contact with infected animals or contaminated animal products, as I previously stated as well, such as, the animal hair industry, the animal meat industry, so et cetera, et cetera. Anthrax is not contagious, so if you touch someone, you breathe on someone, you're not going to get the disease. Anthrax is predominantly found in agricultural regions of developing countries. So in the United States and in Canada and in parts of Europe, you're not going to really find anthrax, especially in the urban cities, but you will most likely find it in the more developing countries such as Argentina, maybe the Caribbeans, in the more agricultural areas. There are actually different types of anthrax for each route of entry, meaning where they enter the body, how they get inside the body, et cetera, et cetera. For cutaneous, that's the most common. It's the one where you get it through abrasions. The spores enter when you get a cut and they land on your skin, they'll enter through the abrasions. It's the least dangerous considering a lot of patients actually come back from it. Unlike the more severe ones, one of the more severe types, and that leads me to pulmonary, or also called inhalational anthrax. It's the most deadly. It's the one that kills many people. It brings them to the terminal stages where it's the most systemic effects. And saying that it can become systemic, this is when the bacterial spores are inhaled and enter through the lungs. Gastrointestinal is the more rare version. It's usually only in developing countries. You don't really see it in developed countries like the United States. This is when the spores enter the gastrointestinal tract or the GI tract after being ingested. And this is usually encountered when someone eats contaminated, undercooked meat from any type of livestock or wild animal. Injection is the most rare. It's the one that's only reported in Northern Europe. And it's usually amongst the drug addicts in Northern Europe, the ones who inject heroin or any other drug into their veins. It enters through the bloodstream, directly into the bloodstream, and it's similar to cutaneous anthrax. The only difference is it spreads throughout the body faster and it's harder to recognize and treat. And as you can see, I have some pictures of the places where you can find anthrax. Also, I have pictures of inhalational anthrax, cutaneous anthrax, and gastrointestinal anthrax. Uh, this is just two pictures for etiology. It's basically a summarization of everything I explained. So with the cow picture, it's the bacteria being found in the soil. The cow is ingesting the plants, which is how it gets into their bodies, and then the bacteria becomes active and they start multiplying and going through the whole process. And then the other one's just a funny picture of all the different types for humans. Moving on to symptomatology. So each type of anthrax infection has common and different symptoms, meaning they have their own unique symptoms, but they also have their commonalities with each other. So for cutaneous anthrax, there's formation of small blisters and bumps that itch, development of a painless sore with a black center, which are usually the black lesions that are around the site of infection, swelling around sore, myalgia, which is basically muscle pain or muscle ache, headache, fever, vomiting, and nausea. Then we go to inhalational anthrax or inhalation anthrax. And these are it has some more severe symptoms, but also some commonalities. So fever and chills, chest discomfort, shortness of breath, tachypnea, which is basically faster breathing, cyanosis, which is basically discoloration of the skin due to low oxygen levels in the bloodstream, confusion or dizziness, cough, nausea, vomiting, stomach pains, headache, diaphoresis, which is excessive sweating in response to the infection, fatigue, body aches, shock, and meningitis, which is basically an inflammation of the fluid and membranes surrounding your brain. Next, we move on to gastrointestinal anthrax, which relates to the GI tract. Fever and chills, swelling of neck and lymph node enlargement, oral bleeding, sore throat, and painful swallowing, dysphagia, which is difficulty swallowing, hoarseness, nausea and vomiting, or hematemesis, which is bloody vomiting, diarrhea and bloody diarrhea, headache, flushing and red eyes, abdominal stomach pain, fainting, swelling of abdomen or stomach. And then we move on to injection anthrax, which is the same as cutaneous anthrax, except because it's going intramuscular, you're going to get abscesses deep under the skin or in the muscle at the injection site. I also have some pictures of cutaneous anthrax, the black lesion, the inhalational anthrax, where the spores go inside into enter the lungs, gastrointestinal anthrax, where you're ingesting contaminated meat, you get in the GI tract, and then also someone injecting the needle, which is injection anthrax. Onto pathogenesis. Systemic anthrax is split into two phases that outline the establishment and rapid progression of infection. This is going to be an overview of the entire process of anthrax disease, and in later slides I will go into depth on each phase. And it's going to begin with the prodromal phase. This phase is the asymptomatic part of the progression of anthrax, and it lasts around 2 to 4 days or longer depending on the individual. It begins when phagocytic cells engulf and transport bacterial spores to lymph nodes. And phagocytic cells are basically cells that ingest or engulf other cells, mainly pathogens. This will lead to phagocytic cells undergoing apoptosis or cell death once they reach the lymph node and unload the bacterial cargo. Then bacterial spores germinate and produce rod-shaped bacteria, or basically final germination bacteria. And this matured bacteria will actually be released into the bloodstream as a result of a lymphatic process. Next comes the fulminant phase. This is the rapidly progressing phase that lasts around 1 to 2 days and ends in the host death, as the pathogen doesn't really take long to kill the individual or the host. Bacteria multiply and spread throughout the body via hematological routes and secrete toxins. And by hematological routes, I'm talking about the bloodstream. And the toxins will actually damage the tissues of nearly all organs. Toxins also disrupt organ function as a result of damaging the tissues of the organ systems and compromise vascular integrity, which causes hemorrhage in microvasculature, which means basically arterioles, venules, small veins, small arteries, capillaries, capillary beds, by affecting blood pressure. Edema also occurs and helps spread the bacteria to other regions of the affected tissues. And edema is basically swelling that allows for the dissemination of the bacteria to other organ systems or other tissues. All of these processes result in death by asphyxiation, or basically suffocation, heart failure, or cerebral meningeal hemorrhage, or basically rupture of the blood vessels in the brain. So, to go into more into depth, the prodromal phase is the first phase in the progression of anthrax, and this phase can be further subdivided into two establishment phases. The first stage encompasses dormant bacterial spores activating as soon as they infiltrate the epithelium of the sites of exposure and are phagocytized by phagocytic cells. And by sites of exposure, I'm talking about the lungs, I'm talking about the skin, the GI tract, or the muscle. The spores will actually begin germinating in this part of this stage in cutaneous, gastrointestinal, and injection anthrax, not inhalational. That is further or later in the process. Activated bacterial spores hijack the cellular mechanisms of phagocytes, namely macrophages and dendritic cells, by secreting an exotoxin. It should be noted that it will secrete two exotoxins, two different ones, that have their own unique effects, but in the case of this specific effect, one certain exotoxin will actually prompt this, and that is edematoxin, or ET. And it's composed of two subunits of protective antigen and edema factor. And the edema factor will actually be the one that causes edema for dissemination and is not cytotoxic. It's not like the other exotoxin, which is going to cause the lethal effects. This one will actually alter phagocytes' behavior of activities such as activation, migration, and production of cytokines. This is also when host cell intoxication begins, because this is when protective antigen binds to anthrax toxin receptors, mainly tumor endothelial marker 8, or TEM8, which is anthrax toxin receptor 1, and capillary morphogenesis gene 2, which is anthrax toxin receptor number 2. These receptors are important because they're actually involved in binding to helping animal cells bind to extracellular matrix and directing endothelial migration and adhesion and a lot of other cellular processes. The spores then are transported from their site of exposure to the mediastinal lymph nodes by phagocytes via lymphatic vessels and to the spleen, or basically through the lymphatic system. And the mediastinal lymph nodes are located in the central chest between the lungs. Spores begin germinating once they arrive at the target lymph nodes in inhalational anthrax. In inhalational anthrax, this is where these spores begin germinating, whereas in the other types of anthrax, they start germinating as they're in the macrophage or the phagocyte that's transporting them to the lymph node. In the second stage, while in a target lymph node, the bacterial spores will actually completely germinate or reach final germination, produce vegetative bacteria, basically bacteria that can proliferate and disseminate and secrete stronger toxins that give stronger effects. And it also replicates during the lymph drainage, or basically when the lymph node is draining lymph back into the bloodstream. An exotoxin secreted by the bacteria will induce programmed lysis of myeloid cells or immune cells immediately after they reach the lymph node, and hemorrhage and necrosis of the lymph node when they fulfill their purposes. So simply put, the exotoxin will kill the myeloid cells or the immune cells as soon as they reach the lymph node. So then they can unload the bacterial cargo and they will not be able to further utilize the immune response. And the hemorrhage of the lymph node basically happens because it's fulfilled its purpose. The bacteria doesn't need it anymore. So it basically ruptures from all the lymph being drained. And then that rupture of the lymph node will actually cause tissue death or necrosis. The myeloid cells most susceptible to the toxin are macrophages and lymphocytes such as neutrophils, dendritic cells, and monocytes. Lethal toxin, or LT, is composed of two subunits of protective antigen and lethal factor. And this lethal factor is what actually kills the lymphocytes and the macrophages. It's what causes necrosis. It basically just, in general, causes lethality. LT inactivates MAP kinase pathways in order to induce apoptosis, while ET will delay apoptosis until phagocytic vehicles transport spores and bacteria to the lymph nodes, or basically it promotes migration. And MAP kinase pathways are mitogen activated protein kinase pathways that are signaling pathways for a group of related proteins called protein kinases. And they play an important role in complex cellular programs like proliferation, differentiation, development, transformation, and namely apoptosis. These toxins will actually create a balance between survival and cell death in order to establish the infection effectively. Because if apoptosis were induced immediately, the bacteria and the spores would not be able to reach the lymph node. So what ET or edemotoxin does is it delays it to allow for the full transportation to occur. And then once it reaches the lymph node, it'll die. And so the bacterial cargo will be able to germinate within the lymph node and the whole process begins. After destroying myeloid cells or immune cells and the lymph node, the immune response is blocked. And it's blocked because all the white blood cells, the leukocytes that are supposed to fight it, are dying immediately once they reach the lymph node. They're not getting a chance to go to the site of infection. They're not getting the chance to transmit signals and complete all their processes. And the dying immune cells will actually release the spores and bacteria to escape the lymphatic system through hematological routes, or basically the bloodstream. Moving on to the fulminant phase. As the second phase in the progression of anthrax, this phase sets the stage for adverse systemic physiology that leads to the death of the host. So this is the phase where a lot of adverse physiological effects actually occur. It starts with bacterial exotoxins accumulating in the blood as bacteria flood hematological routes while traveling to target organs and tissues. So as bacteria are drained into the bloodstream from the lymph node, they will secrete their exotoxins, and those exotoxins will accumulate to very high levels in the bloodstream. And bacteria will quickly proliferate and disseminate until they reach a critical threshold level. As I said, they will reach very high levels in the bloodstream. Bacteria act primarily on the cardiovascular system to kill the host, although bacteria will also act on systems such as the immune system, the nervous system, and the endocrine system. But primarily in this phase, it's going to be about the cardiovascular system. This is how they're going to kill the host, kill the individual. Through direct actions, bacterial exotoxins work to breach the vascular endothelial barrier between the bloodstream and tissues. This is going to allow them to get in between the tissues and the bloodstream and establish their infection. Secreted exotoxins decrease vascular barrier integrity and function of vascular endothelial cells, or VEC for short. Edemotoxin causes hypotension by increasing vascular permeability and inducing vasodilation. So this increase in vascular permeability will allow the toxins to penetrate through the vascularized tissues and induce their effects. And this is basically breaking down the vascular barrier function. This is causing a lot of the dysfunctional effects. Edemotoxin does this by breaching and weakening cell-to-cell junctions. And by cell-to-cell junctions, I'm talking about tight junctions and adherence junctions. Tight junctions are basically the junctions that regulate intercellular movement, and adherence junctions actually regulate extracellular adhesion. It also influences signal transduction, basically through the pathways like the MAP kinase pathways for their transmitting signals. It shifts cell polarity, which is basically the spatial difference in structure, function within the cell. And for paracellular permeability, it's basically the diffusion of ions or molecules via the intercellular space, driven by electrochemical gradients. And then transcytosis is basically transcellular transport in which various macromolecules or any molecules are transported across the interior of the cell. And these are very important processes. They're very important. Edemotoxin and lethal toxins synergistically cause vascular leakage by interfering with the endocytic recycling process and by increasing levels, duration, and production of cyclic adenosine monophosphate, or CAMP, or CAMP for short. And endocytic recycling is basically cells internalizing extracellular material. This removal of membrane obviously has to be balanced, so it goes through endocytic recycling. And this brings back all the removed plasma membrane will come back and be recycled basically. And CAMP, well, that's also important because it's used for intracellular signal transduction in biological processes such as metabolism, gene regulation, regulation of neurotransmitter synthesis, growth factors, and especially immune function, namely the immune response. Edemotoxin alters adenyl cyclase regulation, or AC regulation for short, and basically adenyl cyclase is the mediator for AMP levels, for CAMP levels. Without control of adenyl cyclase, concentrations of CAMP elevate in host cell and cannot be mediated. So when it cannot control the levels of CAMP in the host cell, that will actually cause a lot of adverse effects. It won't be able to regulate the amount of CAMP in the cell, in the blood, and that can cause a lot of catastrophe in terms of signal transduction from the pathways and even other things. High CAMP levels cause endothelial permeability dysfunction, for basically the dysfunctionality of the endothelial barrier. And it also increases cardiac inotropy, chonotropy, and dramotropy. Inotropy is basically a contractile processes. Chonotropy is basically a heart rate, and dramotropy is electrical impulses to the heart. Lethal toxin compromises cardiac structure and performance after the fact, after they're able to permeate the vascular endothelial barrier. As bacteria impair vascular barrier integrity and function, they're able to penetrate through their highly vascularized membranes and disrupt normal organ function. So like I said, as they degrade the function and integrity of the vascular endothelial barrier, they'll be able to penetrate induce their effects, and disrupt normal organ function. This leads to sepsis, or basically shock in the body, and the death of the host by subsequent adverse outcomes such as vascular leakage induced respiratory shock, brain hemorrhage, or disruption of autonomic central nervous system functions. So this is basically a picture or a summarization of anthrax pathogenesis. It is for cutaneous, gastrointestinal, and inhalational anthrax. And as I said in the previous slides, they gain access through the sites of exposure. They are get inside of macrophages or any phagocyte. They travel to the lymph node. They're drained out of the lymph node after there's final germination and production of bacteria. And then they go right into the bloodstream as they're drained directly into it. Now for pathophysiology. We're going to start with the immunological effects, which are the effects on the immune system, and we're going to begin with chemotaxis. Chemotaxis is basically movement of a motile cell or organism, or part of one, in a direction. Now for neutrophils. Lethal toxin and edamatoxin synergistically inhibit neutrophil chemotaxis by reducing formation of F-actin. F-actin is very important. It's a crucial protein for cellular function, such as cell stability, morphogenesis, cell division, endocytosis, and cell migration. And it's also important for motility, or the ability for a cell to move, the movement of a cell. This reduction in formation of F-actin will actually reduce random and direct neutrophil motility toward chemoattractants. Chemoattractants are basically a substance which attracts motile cells, and random motility is spontaneous movement of a cell, whereas direct motility is the direct movement towards the source for a cell. And these are both reduced as a result of the reduction in formation of F-actin. Following that is a reduction in neutrophil polarization, or the polarization of leading and lagging edges of the cell. Now, leading edges are basically the forward movement of neutrophils in the plasma membrane, and lagging edges are the rear movement of neutrophils in the plasma membrane. Edamatoxin specifically reduces the expression of integrants needed for neutrophil adhesion and extravasation. Now, integrants are the principal receptors used by animal cells to bind to extracellular matrix. And neutrophil adhesion is basically the neutrophil adhering to the wall of the inflammation and blocking the adhesive forces of blood flow and other adhesive factors. And neutrophil extravasation is basically the movement of leukocytes out of the circulatory system and towards the site of tissue damage or infection. Now, for macrophages, the exotoxins actually reduce phosphorylation of ERK. Now, ERK is one of the pathways in the MAPK pathways, and ERK is basically the on and off switch for downstream signaling or signal communication in the MAPK and ERK pathway to downstream effectors. Now, downstream signaling is basically a series of events that result in the transfer of a signal, and downstream effectors are stuff like transcription factors and other protein kinases. Hindering the phosphorylation of ERK leads to blockage of downstream effectors such as the major transcription factor of cyclic AMP response element binding protein, or in short CREB, as well as inhibition of its own contributions to cellular processes. ERK is very important when it comes to integrating external signals from the presence of mitogens, protein kinases, such as epidermal growth factor, into signaling events, promoting cell growth and proliferation. And when it comes to CREB, that's a transcription factor that regulates diverse cellular responses such as proliferation, survival, and differentiation. Exotoxins also increase cell migration of bone marrow-derived macrophages, which is detrimental and causes decreased proliferation and differentiation of cells. As for dendritic cells, it just increases chemotaxis and invasion capabilities. And the reason for this is because when dendritic cells ingest the pathogen needs a vehicle that can evade the immune response and all its activities, and also invade the tissues it wants to establish and progress its infection in. More in immunological effects is bactericidal activity. So edamatoxin reduces phagocytosis function of neutrophils, which is one of their main functions, engulfing and ingesting other cells, mainly pathogens. Edamatoxin and lethal toxin influence the oxidative burst of neutrophils, another one of its main functions, and it basically releases reactive oxygen species that will kill pathogens as well as have collateral damage on host tissues. So what ET and LT do is they localize the blast radius and increase or decrease the strength of the oxidative burst, basically manipulating the mechanism of neutrophils. Exotoxins reduce the amount of pro-inflammatory cytokines and chemokines in MB4 cells and dendritic cells. Now, MB4 cells are basically cells that induce maturation of neutrophils. And we already know what dendritic cells are. Now, pro-inflammatory cytokines are cytokines that favor inflammation, and chemokines are what attract white blood cells to the sites of infection. Now, for dendritic cells, they may be affected differently depending on the subtype. So depending on where it comes from, the site of exposure, they will be affected and used in different ways. Exotoxins decrease dendritic cell-mediated stimulation of T and B cells. And T and B cells are very important when it comes to the innate and adaptive immune system. Specifically, etomatoxin alters the development and activation of T cells. Now, this is important when it comes to B cell activation, and when they're deactivated by etomatoxin and their development is interfered with, that will actually inhibit antigen presentation and T cell signaling, which will then obstruct activation of B cells and hinder development of natural killer cells, which help to fight infections. LT, or lethal toxin, causes B cell dysfunction, which, that dysfunction actually diminishes the capacity to produce IgG and IgM antibodies. And these antibodies are very important when it comes to innate and adaptive immunity or immune responses. So because it's diminishing the capacity for this and inhibiting antigen presentation and activation of B cells, this will render innate and adaptive immune responses impossible on the pathogen. It also decreases production of interferon gamma by affecting natural killer T cells, and natural killer T cells are basically a rare lineage of natural killer cells from the T cell lineage. And when it comes to interferon gamma, that's a very important Interferon gamma is basically a cytokine critical to both innate and adaptive immunity. And it functions as like the primary activator of macrophages, in addition to stimulating natural killer cells and neutrophils. For apoptosis, lethal toxin is cytotoxic to endothelial cells. And this susceptibility is what makes endothelial cells more prone to apoptosis, to cell death. Lethal toxin affects the activity of caspases, which are basically proteases or enzymes that break down proteins and peptides that cleave a range of substrates, including downstream caspases, other ones, nuclear proteins, plasma membrane proteins, mitochondrial proteins, which ultimately lead to cell death. And when lethal toxin affects its activity, it'll actually increase the amplification of apoptotic signals and events and allow the exotoxins to control apoptosis. Lethal toxin inhibits the MAP kinase pathways in endothelial cells, as said beforehand. This includes the ERK, P38, and JNK/SAPK pathways. So now this is just a picture of how it affects the different types of cells: macrophages, T cells, B cells, dendritic cells. You can see how the exotoxins induce their effects and how it affects the pathways, apoptosis, reactive oxygen species, and other parts of the processes. Continuing on with cardiovascular effects, it's going to affect the vasculature and cardiac tissue. For vasculature, lethal toxin affects vascular endothelial growth factor, or VEGF for short, the pathways. This prevents proper regulation of endothelial growth, proliferation, and permeability. And this is very important when it comes to working against a pathogen. Etomatoxin rapidly increases vascular permeability, as said beforehand. It does this by interfering with endocytic recycling pathways, reducing cell-to-cell interaction, weakening cell-to-cell adhesion, and disrupting intercellular transport, or transcytosis. Vascular leakage causes cardiac edema. So when these toxins are able to permeate the vascular endothelial barrier, they will cause leakage. And this is what causes cardiac edema. This is what causes swelling of the cardiac tissues. Exotoxins counteract the hyperpermeable effect of VECs, or vascular endothelial cells, evoked by inflammatory lipid mediators in the body. Now, uh, these inflammatory lipid mediators are actually potent enhancers of innate and adaptive immunity activity. Now, when this leakage happens, it causes severely low blood volume, or hypovolemia, and it also causes an influx of calcium ions from because of or in contribution or in assistance of CAMP levels, which causes intense smooth muscle contractions. This leads to vessel collapse and microvasculature, like I said, arterioles, venules, small veins, small arteries, capillaries, capillary beds. And this is what causes severely low blood pressure or hypotension. Following that is narrowing of the outflow tract of the heart, which occurs and causes cessation of blood flow, or basically complete stoppage of blood flow. Moving on to the cardiac tissue, lethal toxin increases the production and concentrations of reactive oxygen species, ROS for short, such as superoxide in the blood. Now, this presence of ROS will actually oxidize myofibrillar proteins such as myosin, actin, troponin, tropomyosin, and it results in cardiac muscle contractile dysfunction, or basically compromised heart contractions. Lethal toxin dangerously decreases systolic and diastolic pressure. Now, when this is affected, it'll increase heart rate. It'll increase heart rate because the heart has to work harder to pump blood. Edema toxin causes myocyte degeneration and myofiber separation, which leads to necrosis. So when the myocytes are not functioning properly and they start degenerating, this will cause separation of the myofiber, which is a bunch of myocytes into one single fiber, one single muscle cell, and this causes tissue death. Synergistically, LT and ET decrease cardiac output, which basically refers to central venous pressure, increased heart rate, and decreased left ventricle ejection, or how much blood volume is being pumped out of the left ventricle. So this is just a picture of the cardiovascular effects. You can see in each type of anthrax that the edema toxin and the lethal toxins start working their way into the vessels and the heart, and they cause hypovolemia, low cardiac output, hypotension, and then death. Lastly, the endocrine and neurological effects. So for the endocrine system, lethal toxin inactivates glucocorticoid receptors. Now, when these receptors are inactivated, this will actually decrease immune activation, because these are mainly for glucocorticoids to transmit signals to activate the immune system. Glucocorticoids released into the bloodstream by the hypothalamus pituitary adrenal axis cannot transmit signals because the receptors are inactivated. And this will allow for inflammation to progress, as cortisol cannot move toward the site of inflammation or infection. Cortisol is very important when it comes to fighting infection. Glucocorticoids cannot mediate the stress response because the receptors are inactivated, and so the inflammation can progress. When this stress response is not mediated, cortisol levels rise and it causes damage to the body over time. And this just adds to the other systemic problems that occur with anthrax disease. All these effects all stem from exotoxins inhibiting the P30 MAPK pathway. Now, for neurological effects, this is mainly going to happen in the blood-brain barrier. So anthrax exotoxins will rapidly cross the blood-brain barrier, which is the cause of meningitis, damage into the brain, and the internalization of pathogen, or the pathogen of anthrax. Lethal toxin changes electrical impulses in the heart, and this affects the cardiac cycle, such as its contractile rhythms and its relaxation rhythms. Lethal toxin affects the function of the phrenic nerve, which is a respiratory stimulus, so it controls the diaphragm and it controls breathing or assists in breathing. Lethal toxin activates the baroreceptors of the sympathetic nervous system, and this causes intense vasodilation or relaxation of blood vessels and a decrease in peripheral vascular resistance, which causes hypotension, low blood pressure. Now, this is just another summarization of everything I talked about, all the effects it has on each system. So on this, you can see on the immune system, chemotaxis is up or down depending on what it entails. Cardiac output is down, glucocorticoid receptor activation is down, phrenic nerve pulse is down, etc., etc. So that's going to be it for my PowerPoint. Thank you for watching. I hope you learned a lot from this, and it was a pleasure being in this class.